Hello, I'm Karen Poe,
And I'm Pia Olafson.
and along with Jesse Evans, Michael Scavarla, and Erica Machtinger, we studied whether shared pathogens could be detected in blood-feeding ectoparasites collected from the same host. One of the main characters in this story is the black-legged tick Ixodes scapularis. This tick is a major pathogen vector and can transmit numerous pathogens that affect both people and animals. As part of this tick life cycle, adult male and female ticks will infest white-tailed deer during the fall to mate and sustain tick populations. However, we find another ectoparasite on deer at around the same time during the fall, and that is the European deer ked, or Lepoptena cervi. These are ectoparasitic flies that will typically infest and bite deer. However, they are also known to bite people. As shown between two papers published by Scavarla and Machtinger in 2019 and Scavarla et al. In 2020, European deer keds are considered invasive and their ranges seem to be expanding, especially further north and west, and this could put more deer and people at risk for deer ked bites. While deer ked bites can cause dermal reactions, the status of deer keds as a pathogen vector is still unknown. However, several bacterial pathogens that are typically found in ticks have been detected in deer keds. Since ticks and deer keds seem to harbor similar pathogens and are found to share the same hosts at around the same time, Poe et al. Also confirmed that deer keds and ticks can be found in similar body regions of white-tailed deer. By being in similar regions, perhaps deer keds can pick up tick-borne pathogens by feeding next to infected ticks through a process known as co-feeding. Since deer keds and ticks share the same hosts, harbor similar pathogens, and are found in the same body regions on the same host, then do they also share the same bacteria and pathogens if they're collected from the same host? And this was a central question to our paper.
So we collected tick and deer ked samples from deer processors and local hunters in Pennsylvania, and the full methods can be accessed using the QR codes at the bottom of the screen. We analyzed black-legged ticks and deer keds co-parasitizing 38 individual white-tailed deer. These were from 23 counties identified in orange, and in total, we screened 306 deer keds and 315 black-legged ticks. We then used molecular assays to target detection of Bartonella species, genera in the family Anaplasmataceae, and Borrelia species. The screening results are presented for black-legged ticks in the left column and deer keds in the right column. Each row represents a different bacteria. Black dots indicate the bacteria was not detected, while orange dots reflect a PCR positive result. So in our study, we found that no Borrelia were detected in deer keds, despite black-legged ticks being positive on the same host, and these are identified by the 10 deer. This is in contrast with previous reports of Borrelia detection in deer keds, and to date, we have not detected Borrelia in the over 600 deer keds that we've screened. Conversely, no Bartonella were detected in black-legged ticks, despite deer keds being positive on the same host, and this was reflected in 32 deer. Again, these are in contrast to prior reports of Bartonella detection from black-legged ticks. Interestingly, shared pathogens were only detected in paired black-legged ticks and deer keds from 3 hosts identified here in orange. Lastly, we detected a rickettsial endosymbiont in all black-legged ticks and observed a higher prevalence of this endosymbiont in deer keds on hosts with paired black-legged ticks versus deer keds on hosts without paired black-legged ticks. In this study, we hypothesized that we would detect similar pathogens in deer keds and ticks co-parasitizing the same host. Our results indicated a limited detection of shared pathogens, and this could be due to limitations of our sampling approach, but could also be due to the different feeding strategy of these ectoparasites, coupled with the season in which we sampled. Overall, our results highlight the need to further study deer ked tick and deer ked host interactions as they relate to the role of deer keds, if any, in the transmission of zoonotic pathogens. We'd like to thank you for your time, and we welcome you to a more in-depth read of our study by scanning the QR code at the bottom of the slide.